In this biocast, we're going to take a look at photosynthesis, the ultimate source of energy for life on our planet. This is because photosynthesis is the process whereby solar energy is transferred into potential energy usable by living organisms. The basic materials in this reaction are carbon dioxide, water, and solar energy. Out of these three things, photosynthesis produces sugars and the waste products, oxygen. Now let's take a look at where this happens. We're going to move the equation out of the way, and we're going to take a look at a chloroplast, which you see represented as this green oval. First thing we're going to do is cut inside of this and take a look at what we have. What we have making our chloroplast are two plasma membranes, an outer plasma membrane shown here, and an inner plasma membrane shown here with our black and gray colors, and then an intermembrane space, this light greenish color here. Now, inside of the chloroplast, in this representation, we see 4 grana. This would be 1 grana here, and 2, 3, and 4. Each of those grana are connected by lamellae, which are represented by these yellow lines here. Now, each one of our granum as well is comprised of 4 thylakoids in this representation. These are the stacked discs. 1, 2, 3, and our 4th thylakoid. And the gray space we see surrounding the thylakoids and the granum and the lamellae is called the stroma. Now, we produce energy by having photons, represented by our little sun here, hit the chloroplast and be absorbed by a granum. Now, the energy of this photon is transferred between granum via the lamellae until it reaches the P680 reaction center. So what we're going to see is our photon come and be absorbed by this granum, transferred between the grana until it reaches this granum, which is our P680 reaction center. So let's see what happens. We get a photon from the sun come, hit the chloroplast, be transferred between our granum until we reach the P680 reaction center. Now, the P680 reaction center is just the granum that's going to take that energy from the sun and start it on its conversion to the sugars that we want in the long run. Okay, so let's dig in a little bit more closely. Now we're going to look at just a thylakoid. So you can see this green circle here represents our thylakoid membrane. So out here we have white, and this is the stroma. And inside here on the gray we have the interior of the thylakoid, just to keep yourself oriented. Now, there are 2 main parts to photosynthesis: the light reactions and the light independent reactions, also called the Calvin cycle. The 2 main reactants in photosynthesis are water, H2O, and carbon dioxide, CO2. When we look at how they move, the water moves through the light reactions, the carbon dioxide moves through the light independent reactions. When the light reactions are working and the water moves through the light reactions, it ends up producing the waste product of oxygen. In contrast, the light independent reactions take the carbon dioxide molecules and end up producing sugars. We need to transfer energy from the light reactions into the light independent reactions because we're taking carbons from our carbon dioxide molecule and we're using them to build a more complex sugar. Therefore, we need energy. And the energy is transferred from the light dependent reactions to the light independent reactions through NADPH and ATP. Both of these molecules are used to store potential energy, and that potential energy then is brought into the light independent reactions and used to convert the carbon dioxide into our sugars. Now, after the potential energy in the ATP and NADPH is depleted in the light-independent reactions, the resultant molecules, NADP plus and ADP, are recycled and sent back from the light-independent reactions back into the light reactions and restored back into ATP and NADPH. So you can see that photosynthesis is an incredibly efficient recycling system as well. Okay, so this was our basics. Remember, we started out with 6 CO2 molecules, 6 water molecules, plus energy, and from that we create 1 complex sugar and some waste molecular oxygen. Now let's take a look in closer detail at how we end up performing this reaction. So, same orientation here. This green circle represents our thylakoid membrane. The white out here represents our stroma, and the gray in here is the interior of the thylakoid. You'll notice we have a bunch of hydrogen ions floating around here. We have something called PS2, which is photosystem 2. We have PS1, which is photosystem 1. We have PQ, which is plastoquinone, and PC, which is plastocyanin. And then we have a few of our molecules that we saw just in the last slide, NADP+. Now we have 2 more complexes here. We have NADP reductase, and remember, since it ends in ASE, it's an enzyme, therefore it's going to catalyze something. Since it's NADP, it's going to be catalyzing the NADP molecule, and since the prefix is reduct, it's going to reduce the NADP molecule. Second complex we have is ATP synthase, again, another enzyme. It's going to catalyze something to do with ATP. Since the prefix is synth, it's going to synthesize an ATP molecule. Now that we know all the players, let's take a look at how they interact. Remember that there were 2 things that needed to get done in the light reactions. We need to split apart a water molecule, and we needed to produce NADPH and ATP. Now, photosystem 2, which you see here, is going to be responsible for splitting apart the water molecule, and photosystem 1 that you see here is going to be responsible for producing that NADPH, and both of them contribute some to producing the ATP. So what's going to happen here in photosynthesis is our photon is going to enter photosystem 2, our water molecule is going to break apart, the oxygen will produce some waste which will leave the cell, the hydrogen ions will contribute to that concentration gradient on the interior of the thylakoid that we see relative to the stroma side. An electron from the splitting of the water molecule will also go into photosystem 2. The energy from that photon will be passed to the electron, so it will energize that electron. That electron then will go through what's called an electron transport chain. And the electron transport chain happens essentially in the thylakoid membrane. It's equivalent to kind of passing down a whole series of waterfalls. Every time that electron takes one more step down the electron transport chain, some energy is given off. Now there's a series of intervening molecules in the electron transport chain, but the only one we're showing right here is plastoquinone. What the electron transport chain does is it provides enough energy so that a hydrogen ion can be pumped from the stroma side into the interior of the thylakoid to further alter the concentration gradient, to make the concentration higher still in the interior of the thylakoid relative to the stroma. After the proton is passed from the stroma side to the interior of the thylakoid, that electron will essentially have lost all of its energy. It will be passed through plastocyanin into photosystem 1, and this time we'll have another photon, this time at the P700 reaction center. The photon will re-energize that electron. It will go through another electron transport chain, providing the energy that NADP reductase needs to take and convert NADP plus into NADPH. The final step of photosynthesis is to make that ATP, remember. What you'll notice is that we've been producing a lot of hydrogen ions on the interior of the thylakoid. So we have many more on the inside than we do on the outside. this creates a gradient, and this is potential energy. As we have the hydrogen ions flow through ATP synthase, that converts the potential energy of the concentration gradient into usable energy, which allows us to take and convert an ADP molecule into ATP. Okay, let's take a look and see how this is actually going to work then. First thing we're going to do is we're going to focus on photosystem 2. A photon is going to hit photosystem 2, the P680 reaction center. A water molecule will split apart. The oxygen goes out as a waste product. The electron goes into photosystem 2. The hydrogen ions stay in the interior of the thylakoid. The energy from that photon will be transferred to the electron. The electron is going to go through an electron transport chain, passing through plastoquinone to plastocyanin. The energy derived from that electron transport chain allowed us to drive another hydrogen ion into the interior of the thylakoid. Now we had plastocyanin pass that electron to photosystem 1, and now we're going to start producing the molecules we need for the light-independent reaction. We're going to need another photon to re-energize the electron that's in photosystem 1. So the photon is going to hit photosystem 1. That's going to energize the electron. It's going to go through an electron transport chain that gives the energy to NADP reductase to convert NADP plus to NADPH, and it's going to go off to our light-independent reactions. The last thing we need to do now, remember, is we still need to make an ATP. We're going to take the ADP and the inorganic phosphate. We're going to covalently bond them together, which will convert the ADP to ATP. But in order for us to do that, we need energy. We're going to create that energy again by the hydrogen ion flowing through the ATP synthase. So we're going to get a hydrogen ion through ATP synthase. The phosphate bonds to the ADP, creating ATP, and it's off to the light-independent reactions. Now the next thing we take a look at will be the light-independent reactions, or the Calvin cycle.